Hey all, my name is Prashanth Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're going to be doing a passage in the Biochem section of the MCAT. You should be taking roughly 8 minutes on this passage, but today, we're going to be going more in depth so you can see exactly what's going on in my head as I take on this section. Let's get it. Alright, great. So I've gone ahead and put the passage on the left and the questions we're going through on the right. So feel free to take a moment and pause the video and come back to us. If not, let's get right into talking strategy. For me, I like to maintain the same strategy going through any passage. The reason for this is I want it to give me something to come back to, regardless of whether the passage is super easy or something super challenging. Having the same strategy and something to come back to gives me confidence, and confidence is so important on test day. For me, the strategy looks like read a paragraph, write a quick 3 to 5 word summary, go on to the next paragraph, and on and on and on. Doing this provides accountability after reading each paragraph, making sure I understand the big picture of what's going on before I go on to the next paragraph. Oftentimes, I'd find myself reading an entire section and then not knowing anything about it, especially when the passages are super challenging. By doing this sort of quick check-in after every single paragraph, I'm making sure I'm accountable for understanding the material before then, and it saves me so much time. I don't have to reread an entire passage, ever. Additionally, the second tip of my strategy is to not spend too much time on figures on that first read. You can give it a quick glance, but I don't want you to spend more time than that. The reason is, we don't know if questions are going to be asking us about the figures or not, right? Why waste time on a figure if we're not going to come back to it? We're already scrapping for time out here, so let's not waste more time than we need. With that, let's get right into this passage. Extracellular phosphate, PI, regulates its own renal excretion by eliciting concentration-dependent secretion of parathyroid hormone. The mechanism by which phosphate is sensed, however, remains unknown. The calcium sensor receptor, CASR, is the main controller of PTH secretion, and researchers have hypothesized that increased phosphate might have an effect on CASR. Alright, so the question we ask ourselves after reading any paragraph is, what's happening? And this is a big picture understanding in simple words. I want you to think as simple as possible because I don't need big words to understand things. I just want a quick summary, something juicy. Alright, so what's happening here? Here, I'm saying CASR controls PTH levels, and the question that they're asking is whether phosphate affects that, right? So I have a little diagram right here, and the question is, does phosphate affect this relationship between CASR and PTH? And with that, I'm done. I have an understanding of what's going on in the paragraph, and I'm ready to go right into that next paragraph. Again, you'll see I wrote arrows, question marks, abbreviations, all of that is perfect. No one else is reading your notes other than you, so why would we spend more time than needed? Alright, let's go on to this. In experiment 1, researchers tested the effect of extracellular phosphate on the ability of CASR to induce PERK, one of its downstream targets. R568 was added to reduce the risk of CA-PI association in the presence of increased PI concentrations. In the study, researchers increased extracellular phosphate levels from 0 micromolar to 2 millimolar. The results are shown in figure 1. Alright, so again, the question we ask is, what is happening? And I want a quick summary. There's so many details that we'll see, but we want something quick. So here I'm just writing experiment 1, right? I'm just going to write phosphate, PI, effect on CASR, PERK, right? That's all we're thinking. What is the experimental setup? The effect of PI on the CASR PERK downstream target. And with that, there's other information, right? The 0 millimolar to 2 millimolar, the results, all of that. And we see the results right here, but we're not going to spend too much time. If we need to come back to it, we will. But right now, we're just looking at the figure. Okay, so it's a Western blot. We're looking at, maybe know that Western blot is proteins. So we're looking at proteins. We're looking at different expression patterns. Other than that, I don't want you to look too much at it. It's just a nice little Western blot. So we can go on to the next paragraph. In experiment 2, researchers measured enzymatic kinetics and demonstrated that PI inhibits CASR in a non-competitive manner. The max response percentage of CASR was measured at increasing CA concentrations in the presence or absence of extracellular pi. The results are shown above, or in figure 2. Okay, so right again, and then we have figure 2, CASR max response percentage as a function of calcium concentration and presence of Pi. All right, so again, what is the question being asked? What's happening? And so what's happening in this, right? And so here we're looking at Ca2 plus effect on response, this max response of CASR, right? And so you can also add plus or minus this extracellular phosphate, right? So this is sort of experiment 2. And so this is sort of a diagram of exactly what we're thinking. And then we look at the figure and we see sort of a graph. And whenever we look at the graph, I just want you to look at the x-axis, the y-axis, maybe the title even. So calcium levels, max response, and then exactly what we're thinking. We just see a plot of max percentage based on these same values we looked at before. And so with that, we finished those paragraphs, and now we're on to paragraph 5. In experiment 3, researchers investigated the effect of Pi on PTH secretion. Increasing levels of Pi were added at a constant calcium concentration, and PTH secretion was measured. The results from the study are shown in figure 3. All right, so again, what is happening? Experiment 3, right? We have another experimental setup. We've looked at two others prior to this, and what are we looking at? We're looking at Pi effect on PTH. That's it. Nothing crazy. If you want, you can write calcium even, so calcium equal or something like that, but you don't have to. And then right away, we see the figure, Pi levels, PTH secretion, exactly what we were thinking. And so with that, that's all experiment 3 is, and we can go down to the final paragraph. Researchers also showed that a mutation of residue R62 in anion binding site 1 abolished phosphate-induced inhibition of CASR. All right, so all we do is write a quick paragraph summary. This is already a short paragraph, so nothing too crazy. But R62 inhibits, right, phosphate-induced inhibition of CASR, right? So this is like probably a longer summary than I usually write, but it's a pretty important sentence to make sure we know, right? This one right here. R62 is abolished, all of that, by this mutation. So with that, we have done our paragraph sort of outline. And with that, we have an understanding of exactly what's going on in this. We have sort of a question that's being posed at the beginning of this relationship between CASR, phosphate, parathyroid hormone. And we have 3 separate experiments that are looking at this. And then we finally summate that into understanding a mutation that is very important into this CASR inhibition mechanism. And with that, let's just start right into the questions, okay? Let's start with question number 1. All right, question number 1. A patient with overactive PTH presents to a clinic. The doctor is most likely to prescribe which of the following hormones? A, epinephrine, B, calcitonin, C, thyroid-stimulating hormone, or D, cortisol. All right, so right away, we should think, what are we looking for? We're looking at parathyroid hormone overactive. So something that pairs with parathyroid hormone to make it normal, right? And so from our content review phase, we should recognize this hormone to be a paired hormone, right? There's an antagonist hormone that works counter to parathyroid hormone. And so we should always think PTH is associated with calcitonin. This should be something we remember very specifically from our content review phase. Why? Because these 2 hormones are very important into maintaining body homeostasis, specifically around calcium levels. And so both of these will affect calcium levels, but how do we remember which one does which, right? So parathyroid increases and calcitonin decreases. And there's a little mnemonic to help you remember that, and that's calcitonin tones down the amount of calcium. So tones down the calcium levels, right? And so with that, we can recognize that this decreases calcium and this increases calcium. And so we have our sort of conjugate pair, and we know what the answer to number 1. But let's go through the other answer choice just so we know what each of these do. So epinephrine that's our classic hormone with fight or flight, right? When we're in a situation, sympathetic nervous system is firing, epinephrine is what's going. C, thyroid stimulating hormone, right? TSH is instrumental in sort of the production of T3 and T4, which are important for metabolism, right? And so this is one of those flat peg hormones, right? And so I want you to remember TSH in the context of flat peg. And I want you to go look that up if you don't remember what that is. And then D is cortisol. And what is cortisol? Cortisol is our stress hormone. So none of these other three answer choices relate to parathyroid hormone. So we can eliminate A, C, and D and left with B2. All right, and so on to question number 2. Question number 2. The researchers include beta-actin levels in figure 1 in order to A, control for cell type, B, control for pH levels, C, control for the amount of protein loaded in the gel, or D, control for unwanted proteins. All right, so it's asking for why we use this hormone or this level of beta-actin in figure 1. So we can take a quick glance at level 1, and we see that our experiment is looking at pi effect on perk, right? So we're mainly looking at perk, but we have this other column or other row for beta-actin, right? And so why do we have beta-actin? Well, usually when we're looking at something that's not really mentioned in the study, it can often be determined to be a control, something we're looking to compare expression levels of some sort to, right? And so this control is exactly what we're using to look at whether we are having a correct experimental setup, whether everything is normal beyond sort of our expression levels. And so beta-actin in this case is a classic example of a control. And so what type of control would that be? Well, we know it's a control. Beta-actin is a special type of sort of molecule that is expressed in all cells, regardless of whether there's an experimental modification, something changed the cell. It's pretty much always going to be measurable. And so why do we use beta-actin as a control is that it's always going to be expressed to a certain level as long as you have cells there. So if we have one cell, we'll have a certain amount of expression of beta-actin. 10 cells will have more expression, 50 cells even more. And so that's what I want you to think of it like. It's an always existing molecule that will be there, that will quantify the number of cells in our sort of blotting. And so if we have the constant sort of measurement of beta-actin across each of these lanes, why are we doing that? Well, we're making sure that each of these lanes gets the same amount of sort of total solution that we can measure, right? If we give like lane 1 sort of like 10 milliliters versus lane 2 50 milliliters, then we'd clearly see that in the beta-actin measurement levels. And so the goal is to make sure that each of these lanes looks identical. And so I'm just going to circle that because we want these to be identical. And that's the purpose of our control. It's a loading control to make sure that everything looks the same beyond the only thing we change in our experimental setup, which is the PI levels, right? This is what we're changing, and this is what we want to see changed. And so we're removing confounding variables in that sense. And so with that, we note that C is our best answer choice because we're controlling for the amount of protein loaded in the gel. We're making sure that each of the cells has the identical amount of beta-actin levels, which quantifies the amount of protein loaded in the first place. And so we're not controlling for cell type because it's always measured in cell type. We're not controlling for pH levels because that's actually mentioned right here, right? That 7.4, 7.4, 7.4, 7.2, so we definitely are not there. And we're not controlling for unwanted proteins. This beta-actin level can only measure for beta-actin, nothing else. So with that, we are able to eliminate A, B, and D, and we're on to question number 3. All right. Question number 3. Lower pH leads to protein denaturation. Which organelle employs a lower pH to denature proteins? Is it A, a protease, B, mitochondria, C, lysosome, or D, nucleus? All right, so right away, we're looking at lower pH organelle denature proteins, right? We're using lower pH levels to denature proteins, and it has to be an organelle. So let's just go through our answer choices right away. So A, protease. Protease is an enzyme. It's not really an organelle, so we can eliminate A pretty quickly. B, mitochondria, powerhouse of the cell. Not really what we associate with denaturing proteins, right? We associate it with metabolism, electron transport chain, energy production, not really denaturing proteins. So we can eliminate B. C, lysosome. This is a good answer. Lysosome is exactly what we think of a molecule of releasing these sort of enzymes to denature some sort of protein because it has these acidic molecules inside of it that bind together with other molecules, bring down the pH, destroy them, denature them, and we're left with denatured proteins. Nucleus, not really associated with that, right? Associated with sort of DNA, the nucleolus, all of that's in there, but not denaturing proteins. And so third standalone question, and we crushed it, right? These are all the types of questions that you need to know. What are we thinking about? Why are we thinking about these different parts of the cell, organelles, and whether they have impact in the way we're thinking? So pretty easy cause and effect type question. All right, question number 4. According to the passage, when researchers add PI to cells, what is the most likely effect on the Km and Vmax of CASR? A, they both stay the same. B, Km stays the same, Vmax decreases. C, Km increases, Vmax decreases. Or D, Km stays the same and Vmax increases. All right, so when we see these terms such as Km and Vmax, right, and we're looking at how pi affects the cells, right, what do we think? We're thinking about enzymes. That's the main thing we're thinking about. We've seen all these sort of tidbits and knowledge from our review phase of the content, and Km and Vmax are exactly what we're thinking about with enzyme inhibitors. And so we have to remember where in the passage we talked about enzyme inhibition. And we are looking exactly right here, right? Where we're looking at enzymatic kinetics and pi inhibiting CASR. And how is pi inhibiting CASR? In a non-competitive manner, all right? So this should be something very important from our content phase, is understanding non-competitive inhibition, right? It's this idea that, sure, we have this enzyme and we have this molecule, the substrate, right? So the red is the enzyme, the black is the substrate, and the goal at the end of the day is for this to bind here. And we're left with one big happy enzyme substrate molecule, right? That's the end goal, is to have this bind together. Well, what happens when we have some sort of inhibitor? Well, we've seen competitive inhibitors, right, in our content review. Competitive inhibitors, we'll use a different color. They're right here, well, it's CI, and they're trying to bind to the exact same spot. And so that is the example of competitive inhibition, right? But now let's go to our next thing. How does non-competitive inhibition work, right? And so non-competitive inhibition has a slightly different sort of structure. Non-competitive has a little attachment on the other side, right? It's an allosteric site, different site from the original site, and it binds to this enzyme to inactivate it. Note that this is not changing the way that the enzyme can bind to the substrate itself. It's changing the way enzyme can be activated by the substrate. So when this non-competitive inhibitor binds to the enzyme or the enzyme substrate, the whole thing doesn't work. And so that's what I want you to know, is that the affinity of the enzyme and the substrate does not change, right? But at the end of the day, the enzyme substrate won't work most of the time, or a lot of the time. And so with these two terms, you should be able to identify what is happening to Km and Vmax, right? So if the ES won't work as much, right, what does that mean? That means that our Vmax, so the peak amount of this binding together of the enzyme and substrate, that will decrease, right? If our enzyme and substrate don't work, right, then our max rate of sort of enzyme substrate binding together and producing this function, that's going to go down, right? Our affinity, so the ability of this enzyme and substrate to match together, right? That's not going to be changed. These enzymes and substrates are fitting together just like they usually are. They're just having this alternative sort of thing binding to it and preventing it from activating. So the affinity, that's going to stay the same. Okay? So again, the affinity, the ability of this enzyme and substrate to bind together, that's going to stay the same. What's going to change is the ability of this enzyme substrate complex to pursue the reaction forward, because this non-competitive inhibitor is binding to the site and inactivating this. And so with that, we should be able to identify that our Km will stay the same while our Vmax will decrease, right? Vmax increases, doesn't make sense. Km increases, Vmax decreases. We know our Km is going to stay the same because the affinity of the enzyme and substrate are going to stay the same. And we know they won't both stay the same because our Vmax will go down if there is an inhibitor involved that is binding to an alternative site. And so with that, we have knocked out question number 4, and we're ready for question number 5. All right, question number 5, our last question. We're 4 for 4. Let's get it. All right. Based on information from the passage, what is the most likely reason that the R62 mutation of CASR abolishes phosphate-induced inhibition of CASR? Is A, R62 is crucial to protein folding? B, R62 interacts with phosphate in the anion binding pocket? Is C, R62 facilitates glutamate interaction with phosphate in the anion binding pocket? Or D, R62 creates the only anion binding pocket in the protein? All right, so right away we're thinking, what paragraph is this in, right? We're hearing R62 mutation, and we should be thinking, oh, I saw this somewhere. Where are we looking? We're looking in this final paragraph, right? Researchers also showed that a mutation of residue R62 in an anion binding site 1 abolished phosphate-induced inhibition of CASR, right? So what's happening? Well, when we change R62, we're getting rid of this inhibition of CASR. So what is R62? Well, whenever we have sort of R or some sort of letter and a number, this is an amino acid, right? This is the location of that residue, and it's sometimes followed with another amino acid that it's changed to. So amino acid start and amino acid end. And so the idea is, in this case, R62 position, the arginine at the 62nd position is changing, right? It's leaving its original spot and becoming something else. And so the idea is, if we're losing an arginine, something is happening. So the question is, what is happening? Well, arginine is a positively charged molecule, right? A positively charged amino acid. It's one of those lists I need you to remember, right? We need to know our amino acids pat. But our arginine is going to be positively charged. And so what's happening is we get rid of that positively charged amino acid, and we're left with no longer any positively charged anythings. And so with that, how does a positively charged molecule interact with the binding site? More often than not, a positive charge is going to be interacting with a negative charge. That's what they're attracted to. Those electrostatic forces. So we're looking, okay, if we're losing a positive charge, that means we should be affecting some sort of binding with a negative charge. All right, so let's go through our answer choice and see if there's any talk of a decreased interaction between this positive and negative charge. All right, A, R62 is crucial to protein folding. That doesn't really make sense, right? Nothing is talking about protein folding. We're talking about sort of a binding interaction at a binding site, right? We're talking about binding site, not protein folding. B, R62 interacts with phosphate in the anion binding pocket. This makes sense, right? What type of charge is phosphate? Phosphate is a negatively charged, right? PO32 minus, phosphate, negatively charged. So we can leave B for now and go on to C. R62 facilitates glutamate interaction with phosphate in the anion binding pocket. Well, we don't really talk about glutamate anywhere else in the passage, right? Why would we just start talking about suggesting a glutamate when in our entire passage we've been talking about phosphates? It doesn't really make sense right here, and so we can eliminate C. D, R62 creates the only anion binding pocket in the protein. Well, we don't know if it's the only anion binding pocket in the protein. We do know that it is an important one because a mutation of R62 gets rid of all of our abolishing of phosphate-induced inhibition. And so since we're talking about phosphate-induced inhibition, we can eliminate D, C, and A, and we're left with B. I think the hardest distinction to make is between this B and C, right? Phosphate versus glutamate interaction, but at the end of the day, we have to decide what are we talking about. We're not talking about any sort of negative charge. We're talking about a negative charge in the context of experiments in which we're dealing with phosphate-induced inhibition, phosphate molecules on parathyroid secretion. It makes sense to talk about phosphate when we're dealing with this. And so we can eliminate C when it's down to B and C, and that is our final answer. We're 5 for 5. Great work, team. All right, great work, team. We crushed another biochem passage. Kudos for you for getting it done. Super challenging stuff, dealing with experimental designs, amino acids, sort of Western blots, all of these sort of concepts that you need to know for the test because they're in that biochem section and they can come up on the biochem or even the chem phys section. If you like this video, give us a like and comment down below exactly what you want to see next. Definitely check out our 528 series playlist on the channel from Vikram. Super helpful tips and tricks and strategies to help you get your maximal score possible. And finally, click on the link in our bio to sign up for our MCAT question of the day so you don't miss out on any practice before test day. Happy studying, and let's get it.